This is David Mount from Brigham and Women's Hospital. My allotted task today was to review the year's development in urate biology and comorbidities. In keeping with GCAN style, I will provide quick sound bites, hopefully soluble, of what I think are some key papers that were published over the last year. These provide a representative mix of papers on uric acid transport, uric acid-associated inflammation, and comorbidities. I apologize in advance to any authors whose otherwise worthy papers have been left out. I have no relevant disclosures. The first paper comes from Owen Woodward in Baltimore and the New Zealand group. This is a truly elegant paper on the effects of the common ABCG2 SNP that leads to a loss of transport function and hyperuricemia. This SNP generates a change in amino acid at codon 141 from glutamine to lysine. Subjects with the lysine allele develop hyperuricemia. Looking at genotyped subjects, the lysine allele is associated with a modest increase in SUA after inosine loading. However, there is no difference in fractional excretion of urate or urinary uric acid excretion. Looking more closely, the wild type or normal uricemic allele, depending on your perspective, I guess, allows for a greater reserve of extrarenal uric acid elimination, which is essentially abolished in the subjects with the hyperuricemic lysine allele on the right. Although these subjects with the lysine allele exhibited a slight increase in renal elimination of uric acid, it isn't sufficient to prevent hyperuricemia. Similar findings were found in the relevant knock-in mouse. This led the authors to propose the following scheme illustrating how this physiology interacts with uric acid loading. Again, this is a very elegant, thought-provoking study. The brief snapshot doesn't do it full justice, and I encourage everyone to search this paper out. The other major genetic influence on uric acid is also, of course, variation in the SLC2A9 gene, which encodes the glutamine urate transporter. This paper came out from my lab 364 days before my talk deadline, so I've included it today. Using a membrane-based yeast-2 hybrid system, which detects interactions at the plasma membrane, we pulled out ITM2B with glutamine as a bait. ITM2B is implicated in hereditary dementias and Alzheimer's disease. Long story short, it potently inhibits the two isoforms of glutamine in both HEK cells and xenopusoocytes. Xenopusoocyte experiments here are shown with both glutamine A and glutamine B. We show that the proteins can be co-IP'd using both tagged constructs and native protein antibodies. This is, I think, an intriguing interaction given the association between uric acid levels and neurodegenerative disease. However, what's relevant to other gout-related stories to come is the surprising inhibition of glutamine N-glycosylation by ITM2B co-expression. That's shown in Western blots in the panel B, where you can see that glutamine A has a lower band in cells that express ITM2B with it. We also see that in the cells that are subject to IP, pulling down glutamine A with ITM2B. Ultimately, we found that this was not linked to the transport effects in that the N-glycosylation mutants of glutamine are fully functional and are inhibited by ITM2B. This observation presages other findings that we've had with other co-expressed proteins that are genetically linked to hyperuricemia. Hopefully, this paper will pave the way for the publication of those findings. This year also saw the identification of two new uric acid transporters. One such transporter was identified by Tony Merriman et al. Through an extension of prior GWAS. These analyses pulled out the OAT7 organic anion transporter, which is liver specific. My lab was able to detect modest butyrate-urate exchange in OAT7 expressing oocytes sensitive to uricosurics. To me, this highlights the potential relevance of hepatic urate transport, which is thus far uncharacterized. This year also witnessed the identification of another urate transporter, GLUT12. The GLUT or SLC2 gene family has three molecular homology classes: GLUT9 is in class 2 along with GLUT5, 7, and 11. We've tested some of these other GLUT2 glutes, class 2 glutes for urate transport, but had never tested GLUT12, which is a class 3 GLUT. Regardless, Dr. Suzuki's group just reported convincing evidence for modest urate transport by GLUT9 in transfected HEK cells, here shown with an EGFP tag construct, which is but the transporter is not affected, plus or minus the EGFP tag. These results lack urate data, and unlike GLUT9, cell depolarization by extracellular K did not activate the transporter. However, SLC GLUT12 does share some other attributes with GLUT9, including evident activation by acidic pH. Uric acid levels in double GLUT12 urate oxidase mice are lower than single oxidase deficient mice, suggesting a role for GLUT12 in mouse uric acid homeostasis. However, there remain many questions regarding the role of this intriguing transporter, not least due to the marked differences in murine and human uric acid homeostasis. Indeed, although the mouse data suggested a hepatic effect of GLUT12, it's not expressed in human liver, nor in human kidney for that matter. These images are from the Protein Atlas database, using GLUT9 in kidney as a relative comparator on the left. Shifting gears to inflammation, this work by the Anders and Steiger group in Munich has been reported previously at GCAN. This published paper is very provocative. It advances the novel hypothesis that soluble uric acid actually suppresses inflammation. The authors used both the hyperuricemic mouse model and human monocytes from control and hyperuricemic patients. These particular figures show attenuation of pouch inflammation by hyperuricemia in this mouse model. The readouts included CD45 positive cell counts on the left, monocyte cell counts, and levels of both IL-1 beta and IL-6. You can see that the hyperuricemic mice had an attenuated inflammation on all 4 readouts. This paper provides an attractive explanation for gout flares after urate lowering therapy, with loss of the associated anti-inflammatory effects of soluble uric acid. Again, clearly a really thought-provoking paper. Finally, moving on to renal comorbidities, June of this year witnessed publication of the CKD FIX and PEARL studies. Prior data had implicated higher levels of SUA and early progression of diabetic nephropathy, as shown in this paper from the group at the Joslin in 2008. Subjects in that paper with the highest tertiles of albuminuria and SUA had the lowest GFRs. PEARL tested whether allopurinol would reduce progression in these diabetic patients. The patients were youngish with an average age of around 50, with modest albuminuria and thus early diabetic nephropathy. After a run-in period to add and optimize RAS inhibitors, they were randomized to 100 to 400 milligrams of allopurinol versus placebo for 3 years. They followed GFR progression with the gold standard direct measurement of GFR with iohexol. Notably, on the left, as you can see, they achieved a significant drop in uric acid, but no effect on the primary outcome, reduction in GFR's progression. The CKD FIX study came out in the same issue of New England Journal of Medicine. These patients were older with more marked albuminuria, but there was a heterogeneous mix of diabetics and non-diabetics. Again, the primary outcome, preventing the reduction in GFR over time, was negative. Some observers pointed to that last upswing of GFR in the allopurinol group on the right, however, suggesting that maybe if the trial had been continued for longer, they would have had a positive result. However, these patients had higher uric acid than in PEARL, that did, however, go down significantly on allopurinol, with in the bottom panel no effect on albuminuria. There have been a lot of criticisms and concerns about these papers. CD FIX is underpowered. Again, there was perhaps that flattening in the GFR curve if it had a longer study. CKD PEARL, the patients were not hyperuricemic per se, and they had type 1 diabetes without substantial proteinuria. However, these really were the sort of patients that were studied previously with early GFR loss in diabetic nephropathy. One other concern was the incidence of death in the allopurinol arms in both studies, here shown for Pearl, where you see 4 in the placebo and 10 in the allopurinol arm. Finally, a paper that appeared last month in JASN provides a potential way forward in looking at renal comorbidity. Again, this study is from the Anders and Steiger group in Munich. They found no potentiation of aristolochic acid nephropathy in mice with hyperuricemia. However, when they added an acidogenic diet to their hyperuricemic model on the upper left, they detected a significant risk of CKD associated with crystalluria and medullary crystal-associated granulomas. Again, this provocative study raises more questions. For example, I typically don't associate CKD with hyperuricemia, so the relevance to hyperuricemic CKD isn't entirely clear. However, this study emphasizes to the nephrology community what rheumatologists have known for years in hyperuricemia is that it's the crystals that do the damage. Thank you very much, and I hope you enjoy the rest of GCAN.